NMR spectra. NMR spectra is usually a graph between strength of magnetic field and intensity of signal. Each absorption peak represents to at least one nuclear species. The isotopes of the elements are spread out into the spectrum on the basis of their differing gyromagnetic ratio. Spectra are generally very simple and give sharp absorption lines. The lower the limit of detectability in terms of numbers of nuclei per centimeter cube is 10 to the power 16 to 10 to the power 20. Thus, NMR spectroscopy is used with concentrated solutions. Line width is affected by the physical state of compound in which the isotope is located, and the height of the peak is determined by such factors as numbers of nuclei contributing to the signal, that is concentration of sample, temperature of the sample, and gyromagnetic ratio. You can see how this spectra comes out. इंटेंसिटी और मैग्नेटिक फील्ड के बीच में स्पेक्ट्रा इस तरह से शार्प पीक्स के फॉर्म में दिखता है. There is one important factor linked to chemical to NMR, which is chemical shift. Now, what is chemical shift? Different molecules, and even within one molecule, electronic configuration of a nucleus will differ slightly, reflecting the difference in chemical bonds. The electronic cloud surrounding the nucleus is modified, and the result is an alteration in the precision frequency, resulting in the displacement of resonance line when the same nucleus is observed in the same external magnetic field but in different types of bonds. This shift in the resonance line is known as chemical shift. फिर से पढ़ ले. The electronic cloud surrounding the nucleus is modified. The electronic cloud surrounding a nucleus is modified, and the result is an alteration in precision frequency, resulting in the displacement of resonance line when the same nucleus is observed in the same external magnetic field but in different types of bonds. This shift in resonance line is known as chemical shift. एक ही न्यूक्लियस है, एक ही एटम है, उसका न्यूक्लियस depending on the bond with which that atom is linked behaves in a different manner when NMR is done. So this is known as chemical shift. Chemical shift in a simple molecule are fairly characteristic and is used for analysis and structure determination of various compounds. Chemical shift जो है वो characteristic feature हो जाती है, तो उसकी वजह से एक कंपाउंड में हाइड्रोजन प्रेजेंट है या एक कंपाउंड में प्रोटॉन्स प्रेजेंट है, तो उसका एक कैरेक्टरिस्टिक वैल्यू होगी, उसकी कैरेक्टरिस्टिक स्पेक्ट्रा आएगा उसका एनएमआर में. लेकिन अगर वही एटम किसी दूसरी बॉन्डिंग में है, किसी दूसरे कंपाउंड में, तो उसका कैरेक्टरिस्टिक स्पेक्ट्रा डिफरेंट आएगा. इसलिए एक कंपाउंड को दूसरे कंपाउंड से केमिकल शिफ्ट की वजह से फर्क किया जा सकता है, जो कि एनएमआर डिटेक्ट करेगा. NMR spectroscopy has been widely used by chemical analysis. There can be no interference from compound of any element other than one under study. तो यहाँ पे इंटरफेरेंस नहीं मिलता कि एक साथ कई कंपाउंड्स हो, लेकिन नहीं, जो हमें देखना है अगर हम उसको देखेंगे तो उसमें कोई फर्क नहीं आएगा, कोई इंटरफेरेंस नहीं कर पाएगा. The technique is widely used for determining the structure of various compounds. For proteins, NMR is useful in following ways. These are the applications. To determine the fraction of amino acids in the alpha helical configuration, it can be used to monitor helix coil transitions and especially to observe local changes during transition. जब helix और coil आपस में बदल रही हैं, sometimes during denaturation also this happens. So ये इसको पता किया जा सकता है to determine the conformation of selected regions of protein around a particular amino acid. NMR can be done to observe the binding of a molecule and metal ions to selected regions of the protein by observing either the ligand or protein spectra. किसी भी स्पेक्ट्रा को देखेंगे, एक पर्टिकुलर साइट को फोकस करेंगे तो वहां पे जो भी चेंजेस हो रहे हैं, मेटल आयन बाइंड हो रहा है, कोई दूसरा लिगेंड बाइंड हो रहा है या हट रहा है, that can be observed by change in spectra in NMR to study the paramagnetic magnetic active sites in electron transfer reactions or proteins. Properties of active site of enzyme may be studied. Effect of cofactor on enzyme substrate binding is studied. Binding of a polymer to protein and the DNA histone complex can be studied. Indication that one of the two histidine is near a particular alanine in an enzyme can be found out. Base pairing structure of transfer RNA may be studied. Distance between binding site and an amino acid residue of a protein may be studied. Now, instrument कैसा बनाया जाता है? Instrumentation. Two types of NMR spectrometers are commonly encountered. They are a continuous wave NMR spectrometer and Fourier transform NMR spectrometer. Here we will study about the first one, continuous wave NMR spectrometer. Just see the figure. These two are the strong magnets. This is sweep coil through which sweep generator है ये. रेडियो फ्रीक्वेंसी के लिए ये कॉइल है, रेडियो फ्रीक्वेंसी जनरेटर के लिए, और स्पिनिंग सैंपल ट्यूब, यहाँ पे स्पिनिंग हो रही है, जो सारे हम अभी डायग्राम्स देख चुके हैं. फिर रेडियो फ्रीक्वेंसी रिसीवर है, एम्पलीफायर है एंड कंट्रोल कंसोल एंड रिकॉर्डर. फाइनली यहाँ से रिकॉर्ड किया जाता है. ठीक है. Resonance frequencies of nuclei in a sample placed in the magnetic field by sweeping the frequency of RF radiation through a given range and directly recording the intensity of absorption as a function of frequency. The spectrum is usually recorded and plotted simultaneously with a recorder synchronized to the frequency of RF source. About the basic components, a radio frequency transmitter generates an appropriate RF to irradiate the sample. Radio frequencies are generated by electronic multiplication of natural frequency of quartz crystal contained in a thermostat block. Different crystal sources and transmitters are used for different resonance frequencies. The radio frequency oscillator coil is installed perpendicular to the magnetic field and transmits a radio wave of fixed frequency such as 60, 100, 2000, 200 or 300 megahertz to a small coil that encircles the sample in the probe. The RF source is associated with a power controlling device so that the level of RF power can be varied and adjusted empirically to get the best response. Field on x-axis is horizontal. The strength of resonance signal is directly proportional to the number of nuclei resonating at that field strength. Computer enhancement, known as computer averaging technique, or CAT, and it is very useful for studying very weak absorbing biological samples. In this technique, the readout from mm, sorry, this is 10 to the power 7 to 10 to the power 8 spectra of sample are superimposed and background noise is minimized. This is the summary of nuclear magnetic resonance. Nuclear magnetic resonance is used to determine the structure of molecules, including small soluble proteins. The word nuclear does not refer to radioactive decay. Nuclear magnetism merely relates to properties of nucleus similar to common magnetism we are familiar with. Nuclear magnetism is 1,000 times weaker per atom than the magnetism associated with electrons. The frequency of radio waves required to cause resonance in different nuclei depend on the way atoms are arranged in a molecule. The analysis of molecules NMR spectrum therefore allows a structural model to build. NMR spectroscopy is used for structural analysis of proteins and other molecules. Key principles: some atomic nuclei are intrinsically magnetic. Such nuclei wobble in magnetic field when exposed to radio wave at certain frequency. This is nuclear magnetic resonance. Nuclear dipoles in the sample align in the magnetic field and can absorb energy and flip from one orientation to another, and later flip back to and re-admit sorry, that energy. The stronger the dipole, the greater the energy associated with the alignment. The sample absorbs some of the radio frequency and after some time re-emits radio signals which are amplified by receiver and stored for interpretation. The frequency of radio waves required for resonance depends on the way atoms are connected together and their relative position in a space. The structure of a small molecule can therefore be deduced from its nuclear magnetic spectrum. Sorry. The nuclei of some atoms behave like tiny bar magnets. Any small magnet will align itself in an external magnetic field. For example, a compass needle will align with the Earth's magnetic field and point north. Atomic nuclei can behave in the same way. If you move compass needle with your finger and force it to point south, it will be unstable. It will soon reorient itself in a magnetic field and point north once again. Similarly, atomic nuclei can be reoriented in a magnetic field by supplying energy in the form of radio waves, but will soon return to the most stable orientation. At just the right frequency of radio waves, a nucleus will flip backwards and forwards between the stable and unstable states. This wobbling effect is called nuclear magnetic resonance. The radio frequency required to achieve magnetic resonance depends on the type of atom, what other atoms it is connected to, and what other atoms are nearby. By analysis of NMR spectrum of a molecule, a graph showing NMR in a molecule over a range of radio wavelengths, it is therefore possible to deduce three-dimensional structure of that molecule. how this is used then. As with the X-ray crystallography, one of the main applications of NMR spectroscopy in biology is to determine the structure of proteins and other molecules. NMR is particularly suitable for the analysis of small soluble proteins. Structures solved by NMR are generally presented as an ensemble of models, all of which satisfy an available data. This is I sent one link to you from YouTube, Nobi. I hope you have heard that lecture from Nobi. If no, then go and find out the link on WhatsApp and read this. With this example, you will understand what is happening in NMR. And this is the example of chemical shift that is also given in the lecture in YouTube. Okay, so this was all for NMR spectroscopy.